So the first part of the talk, which I promised to complete in this presentation, will be purely a computational work, and all carried out by Farhan, a graduate student. And all this work that we do is funded by core funding from DAE through TIFR and NCPS, and we also have a grant from DBT, Welcome India Alliance. And if I get to it, the second part of this talk, that work was funded by a DBT grant. Okay. So bacteria adapt to changing environments, and adaptation is something that spans a very wide range of timescales, from the matter of within the cell generation, bacteria adapt to their circumstances by changing their gene expression states, and bacteria have been around for a couple of billion years, and they have there is a huge phenotypic and genotypic diversity of bacteria on this planet, and I think they estimate that there are probably over a billion bacterial species. And one of the most dramatic demonstrations of bacterial adaptation at fairly short time scales to changing environments would be antibiotic resistance, and this is genetic evolution. So under selection, bacteria change their genotypes to achieve new phenotypes, which, for example, in this case would be the ability to resist an antibiotic. And in the lab, there have been some very interesting demonstrations, and of which I will talk about this famous study from Richard Lenski's lab. He started evolving E. coli in laboratory conditions sometime in the 1980s. So he takes an inoculum of E. coli, puts it into fresh media, lets it go into stationary phase, and then dilute it back into fresh media. Keep doing this every day, and he's been doing this for now 30 years plus. And starting from whenever the 1980s that he began this experiment, he's now completed 60,000 plus generations of E. coli. And along the way, a variety of new phenotypes were discovered. For example, E. coli, which was not previously known to have the ability to utilize the metabolite citrate as a carbon source, evolved that ability, for example. And now these sequences are available, and various phenotypes have been described. And this is genetic evolution, and at a much rapid time scale. That's again within a single cell. Within a single generation, you have gene expression changes. You have which is primarily driven by proteins called transcription factors. Transcription factors are proteins which would bind upstream of a gene and either recruit the RNA polymerase or block access of the DNA to the RNA polymerase. So these transcription factors can be activators or repressors. The activity of these transcription factors in turn would be governed by their environmental conditions. For example, some of these transcription factors might directly bind to a small molecule signal, and this binding may determine whether these transcription factors are active or not. And in other cases, these transcription factors may be phosphorylated by other signal sensing proteins. So there's a diversity of ways by which these transcription factors can be activated by the environment, and then subsequent to their activation, they play a role in activating or repressing the expression of specific genes. And unlike genetic evolution, which requires multiple generations to take effect, these gene expression changes are temporary and will operate within a single cell generation. Now, these transcription factors and their target genes form what we refer to as transcriptional regulatory network. So E. coli has about 300 transcription factors, of which we know at least one target for under 200, and many of these transcription factors have a very few targets, one or a few targets, and these transcription factors specifically respond to one signal and result in gene expression changes that can specifically respond to that signal. And then the second type of transcription factors are what are called global regulators. And these global regulators can respond to a whole range of parameters and can regulate hundreds of genes. And as one would expect, these global regulators are also expressed in the cells at much higher levels than these local on-demand regulators. So, how does genetic evolution and gene expression change? How do these talk to each other? So that's the question. For example, how would adaptation proceed, for example, by mutations in transcription factors, thus affecting gene expression states? So when we talk about regulatory networks, we can look at this at multiple levels. One, there is the transcription factor itself, and two, there are the binding sites to which these transcription factors bind. Much of the literature that is there favors looking at transcription factors, at least in bacterial systems. Primarily because if you're doing this computationally, it's fairly straightforward to identify transcription factors from their protein sequences. But it's much more difficult to identify these binding sites, and these predictions tend to have fairly high false positive and false negative rates, because especially for these bacterial transcription factors, outside of a few outside of several transcription factors in E. coli and a few in certain model organisms, we just don't have enough data to understand these. So a lot of the work that we have has focused on how transcription factors evolve. And what I will be talking about will also focus on transcription factors themselves. In the past, a lot of the analysis of this kind is involved looking for a transcription factor, say in E. coli, and see if orthologous proteins are found in distantly related bacterial species. So what these studies have looked at is how the repertoire of transcription factors would change from one bacterium to another. Now, way back in 2004, Madan Babu and Sarah Teichmann suggested that a lot of these transcription factors they arise by gene duplication. You have one transcription factor, and then over time these duplicate, and then you have very large transcription factor family. So this is one evolutionary force which is suggested. And then later, Adam Arkin's lab said, well, it's we don't fully agree with this view. We think most of these transcription factors are horizontally acquired. And especially if you look at this box, which is about neighbor regulators and so on, you see that a large proportion of transcription factors are predicted to be horizontally acquired. In contrast to global regulators of transcription, many of which are predicted to be native. Now, again, here we are really talking about gains and losses of transcription factors across fairly large evolutionary distances. Now, can we also have situations where transcription factors basically evolve by just point variations, and what is the contribution of these point variations to transcription, to adaptation at short time scales, and probably even to the variation in these transcription factor repertoire across much longer time scales? There is evidence that this is happening. Again, if this is again from Adam Arkin's group, this figure on the left. This is a comparison between two very, very divergent bacteria for which some amount of regulatory network information is available. This is E. coli and Bacillus. So they were comparing orthologs of transcription factors and their targets between these two organisms. And the conclusion that they came up with that you might have both the transcription factor and the target conserved, but the regulatory interaction itself may not be conserved, which suggests that you're not really looking at gains or losses of transcription factors and their targets, but variations in the sequence of these proteins and the binding site DNA, which might be causing these changes. And there are also experimental studies looking at selected transcription factors in different organisms, and which is suggested that you can have a transcription factor which may be able to bind to a DNA in one organism, but may not be able to bind to the orthologous binding site in another organism, and in other cases where it can bind but it cannot recruit the RNA polymerase and activate transcription, again suggesting the roles of point mutations. And this study again by Gauri Shankar's lab here in Hyderabad. So he showed that you can have amino acid substitution variants of a transcription factor in one organism can activate a distantly orthologous target. Now, for all of these, looking at point variations, we have individual examples such as the studies mentioned on the right, and some limited comparisons at very, very long evolutionary distances. But this is not something that has been studied in very systematically. So we decided, okay, let's ask this question: how have how do and how will transcription factors change over time? So it's an interesting question to ask. Now, and this part was also inspired by again this study from Madan, where he again looked at orthologs. So this goes back to these kinds of studies where one is just looking at orthologs of transcription factors and non-transcription factors and seeing how well they are conserved or not. And whatever, so he's in this study, he's taken all transcription factors in E. coli, all non-transcription factor target genes again in E. coli, and then here's these dots. There are many of these dots. Each of these dots is an organism in which the presence or absence of these transcription factors and the target genes are being looked at. Now and then it says, okay, here is one organism where 10% of the transcription factors have orthologs, but close to 20% of the target genes have an ortholog. And then does this for a couple of hundred bacterial genomes. And then the conclusion that they arrive at is that transcription factors are less conserved than non-transcription factor target genes. Now, the issue with this is that transcription factors and target genes are not fully independent entities. There are certain transcription factors would regulate a subset of target genes. So it might now be worth comparing transcription factors with their own target genes as opposed to bunching all transcription factors together in one basket and all target genes in another basket. But that apart, the conclusion from this is that transcription factors are less well conserved than non-transcription factors. And this is also something which is done across 175 bacterial genomes from different phylogenetic groups, and so we would expect this to represent a billion years of evolution. Now, what we wanted to do is again to now start focusing on this at a more a limited time scale. So let's now just look at one species, that's E. coli, and take advantage of the vast variety of genomic data that are now available for any given species of interest. For example, if you look at NCBI short read archive for genomes of E. coli, probably will there were about 40, 50,000 a couple of years ago. So there'll be many more. Of course, there'll be a lot of redundancy and so on. But this will certainly provide a substrate for us to look at evolution within E. coli in some detail. So again, this is 90,000 hits last year. So it's quite a large number of genomes that are available just for E. coli. So we just set a few thresholds that you should there should be at least a reasonable coverage, and we also insisted that these should be paired end reads and so on. And then ended up with about 20,000 sequencing runs that were there. And then what we did is to basically remove the redundancy based on some kind of a clustering method, so that we using only genomes which are reasonably well spread out within the E. coli phylogeny. And once we have this set of genomes, which finally came down to about 2,500 or so. And with these 2,500 genomes, we can estimate sequence diversity at synonymous sites, non-synonymous sites, and so on. We realized that the sequence diversity in synonymous sites for some reason seems to correlate with gene length, even when it is normalized for, even if this is at a per site level. We don't know why that is. So, but non-synonymous diversity doesn't. So what I'll be talking about is non-synonymous diversity of transcription factors and target genes. So once we have this measure, we can calculate these measures specifically for transcription factors and their target genes. And since there is a chance that there may be some systematic variation in genetic diversity estimates from one study to the other, we normalize transcription these sequence diversities that we get by the sequence by the average sequence diversity of these non-transcription factor target genes within that data set, so that across data sets we are on more or less the same footing. Now, what we find is the following. So these are 15 different data sets that produce these 2,000 plus sequences. These are along the x-axis. And on the y-axis is the difference in sequence diversity between transcription factor and its own target gene. So we are not bunching all transcription factors into one pool and all non-transcription factor target genes in one pool. Instead, we're taking a transcription factor and its and the average diversity of its target genes. And what we are seeing here is that within E. coli, transcription factors seem to show less variation than their target genes, which is seems to be in contrast with these other studies that I referred to, which mention that transcription factors are less conserved than their target genes. Yes.
Can I interrupt you? Sorry.
Yes, please. Yes, please.
So my question is like, so when you compare their target genes, so are we talking about the putative promoter regions of this target genes?
Oh no, the
gene body.
The gene body. Okay. So we're just comparing protein with protein. There's a non-synonymous sequence diversity of the transcription factor gene and the non-synonymous sequence diversity of the target.
Got it. Thank you.
Right. So within E. coli species, transcription factors show less sequence variation than their target genes. As I had mentioned earlier, there are transcription factors which have only a few targets, and there are those which have many targets. Transcription factors which regulate a few targets have a fairly small range of influence, and so anything that affects them may affect only a few genes downstream. And whereas a transcription factor that has a large number of targets, if there is a mutation which affects its function, then a lot of things can go crazy. So, how does the sequence diversity of a transcription factor vary with this range of influence? And what we see is that these influential transcription factors seem to show fewer variations than local transcription factors. So, taken together, this suggests that transcription factors which may have pleiotropic effects seem to show fewer variations than transcription factors which may have limited effects, though any transcription factor mutation is likely to have an effect on more than a few genes. So many of them will have a few targets. So even local transcription factors have an effect which is show a sequence diversity which is less than their targets. So from these previous literature, we see that across bacteria, transcription factors tend to be more gained or lost than non-transcription factors. But within E. coli, we see the opposite, that transcription factors show less sequence variation than their target genes. Okay. So this is a recap again, that transcription factors are less conserved than non-transcription factors. And as I had mentioned earlier, this doesn't compare transcription factors with their own non-transcription factor target genes, but just puts all of these into one basket, which can be problematic. So we wanted to look at that as well. And another change that we thought we would do is, still, when we're looking for orthologs as well, not to go across all bacteria, because after a point, identifying orthologs so on also can become a little messy. So we decided we'll stick to within the clade of gamma proteobacteria. And the first thing we asked is conservation of a transcription factor across species. How does it correlate with sequence diversity within a species? That is, and what we find here is that a transcription factor which shows high genetic diversity within a species tends to be more gained and tends to be more lost in other gamma proteobacterial species. So again, this would say that transcription factors tend to show less sequence diversity, but these previous results suggest that transcription factors are more gained or lost across species. How do these reconcile? And what we do see here is that there are many transcription factors which, in fact, have very low diversity within the species, but again fairly low conservation across species, which seems to nicely tie in with the observations from Adam Arkin's lab, that a lot of this gene gain and loss can be due to horizontal gene transfer, and this need not really correlate with the extent of sequence variation that one might see at shorter time scales. And as I had mentioned, a small point here is that the previous studies which looked at transcription factors and target genes, again, did not pair transcription factors with their target genes, and instead pooled transcription factors in one basket, target genes in one other basket. Now, if we were to do this pairing between transcription factors and their target genes, we really do not see much of a difference across gamma proteobacterial genomes. And again, we do see, similar to what we had observed for sequence variation within the species, even across gamma proteobacteria, we again find that specific tiers with a small range of influence are less conserved across these species, which is again consistent with Adam Arkin's horizontal transfer idea, limited to local transcription factors and not to global transcription factors. So at this point, what we have is that sequence diversity over short timescales correlates partly with a gene gain or loss across species. And uh, transcription factors with low sequence diversity within a species can be gained or lost across species, and which may be consistent with their horizontal gene transfer. And again, within a species, transcription factors tend to show less sequence diversity than non-transcription factor genes. Okay, so now we have this bunch of observations. So can we try and understand this a little better with possibly with some kind of time course information? And for that, we went and looked at Richard Lenski's experiment that I had mentioned at the beginning, where E. coli had been evolved for 60,000 odd generations, and recently they had sequenced genomes over time. And we in this context, various other laboratory evolution experiments have shown that early adaptation to a new condition seems to involve a variation in some transcription factors early on. So, but we just wanted to look at this more systematically, and for that, Richard Lenski's time course was a good study. So again, here is a timeline. So, well, 1988 is zeroth generation. 2016 is 65,000 generations. And here, as you can see, that they've marked various landmarks in their experiment that they had observed. I'm sure there are many more. So we can now measure some. We can get some measure of sequence variation over time. Now, what we have here is basically, these are population genomes, which is sequencing a genetically heterogeneous population. And when we have these kinds of population, we're just measuring the frequency of mutations in a protein. And this is something that we can look at target genes, that is non-transcription factors, that are TGs, LR for local regulators with a small range of influence. These pink purple ones are global regulators with large number of targets. And what we're seeing here is that these transcription factors seem to accumulate a lot more mutations than target genes. So that's, for example, as you can see, these pink and yellow lines seem to have accumulate much more mutations than target genes. The target genes are this very, very low slope line, which is at this point. And this is and these are accumulating these mutations very rapidly at early time points, and then there seems to be an overall decline. And again, these measures are a combination of new mutations coming in as de novo mutations, and plus change in frequency of existing mutations. And what we wanted to look at here primarily are new mutations that emerge. So if we take that out separately, we see here that the de novo mutation frequency of these transcription factors, again the same colors, these yellow and pink ones are transcription factors, green are target genes, and that these transcription factors have much higher rate of de novo mutation frequencies early on, and then they also decline much faster. And if one would actually zoom in here, I don't have that picture, is that at the end of these 60,000 generations or so, the rate of new mutation accumulation in these transcription factors seems to have gone below that for non-transcription factor target genes. And you don't see these kinds of very strong patterns in these mutated lines, which just mutate a lot. And the real difference between these two is that one can expect many of the mutations in non-mutators to be adaptive, and most mutations in these mutators to be neutral. And whereas the difference between transcription factors and target genes at early time points is significant, as indicated by some of these blue dots in the non-mutated lines, it is not significant in these mutators. So, at very, very short time scales, as a bacterium is adapting to a new environment, there seems to be de novo mutations accumulating in transcription factors, and then these decline to a level which is less than what we would expect for even target genes. And then when we just take a jump and move to about 100 million years of E. coli's divergence, we see that transcription factors in fact show smaller sequence diversity than their target genes. And some of that values in terms of their sequence diversity carries over to at much longer phylogenetic distances of, say, within gamma proteobacteria. And there is also evidence for transcription factors which don't really change much in sequence within the species, but are extensively lost in other species, pointing to a role for horizontal gene transfer at these time scales. So putting these together over short time scales, initial adaptation again seems to proceed via these mutations in transcription factors. But over time, there may be other mutations which come in these target genes, which can be more precise and also override the selective advantage that is given by a pleiotropic mutation in transcription factors. So over time, you see that transcription factors show less sequence variation than non-transcription factor target genes. And over much larger phylogenetic distances, where presumably greater habitat and other types of niche diversification effects come in, factors such as horizontal gene transfer would result in bacteria having entirely different repertoire of transcription factors. So again, early adaptation is driven by regulatory mutations in many cases, and over time, very likely negative selection against pleiotropic mutations decreases these regulatory mutations, which may have large effects. And then over much longer time scales, we have gene gain and loss processes changing the whole repertoire of transcription factors. Okay, so that's part one. Are there any questions here before I move on to part two?
Ashwin, I have another question. So,
Yes.
even with the mammalian systems, mutations are something like which basically leads to many predictions, like correlations with X, Y, and Z parameters. People have shown, they correlated mutations with n number of things. So, my question is, since I am not aware of the literature, since genetic manipulation in E. coli is very straightforward, like homologous recombinations, it can be done in 3 days, no? So my question is, like, even with Linsky's experiment, where he has done the first round and then the last round, so people have also done some sort of retrograde mapping, like, you get the isolate from the
Correct.
like
Yes.
100 200 generations, and then try to induce mutations specifically at the loci, so
Yes.
that they can make the features of the older ones. Is some sort of experiment has been done?
So so these are so in fact the second part I'll talk about some more experimental work as well. So in E. coli, if you're talking about any specific study and talking about specific mutations, then it's one has to systematically show that there that it has a specific effect, and how, and it's also fairly straightforward, though well time consuming and all of the other challenges are there, to in fact look even at things like epistasis and so on if we have a hypothesis. The problem with any experimental study when one is moving reconstructing strains is that we don't always know what effect a genetic background might have on something. So, which means that any of these studies will become fairly long drawn.
I got
And
it. Your point is like in the epigenetics, people say there is these regions are CPG islands, and they
yeah,
show methylation here, demethylation there, and
yeah,
they correlate with the expression, But
right,
not even a single paper, at least to my knowledge, I've ever come across where they have manipulated the CPG island, not with the mutations, but with the methylation itself, and they show the effect. It's all
yeah.
correlative. So I think something in this regards of the, like, the evolutionary mapping of the TF binding and TF efficiency or whatsoever. So some sort of proof of principle experiment is required, you know.
So yeah, so with E. coli we have quite a bit. E. coli yeasts and so on. I think there lot of experimental literature, but I think the methylation is also a little bit more complicated to do it in the cell, right? Because it's not always straightforward
So that I
to
used as an example. That I
right.
used
that's
as an
that's
example.
that's especially complicated to do even with E. coli, right? How do you specifically change methylation only at that site, unless you're doing it in vitro or something like that. Um, so that so I think even with E. coli, that kind of evidence is there where you can look at methylated sites versus non-methylated and so on. But yeah, there are experimental work with a lot of good manipulation studies happen only with model organisms. With in eukaryotes, you'd see yeasts, flies, and so on, where there would be a lot of studies of specific mutations. But I think it just gets a little messy with mammalian systems. That much harder.
Okay, I think I got my answer. So my second question is, when you basically compared the DNDS analysis when you did, so you took
Yeah.
into account the TFs and the gene bodies, like the protein coding genes of their targets. So
Yes.
and you basically found some sort of effect in the TFs compared to their protein coding counterparts, like their targets.
That
So
is the targets that are affected by this transcription factor G.
exactly,
Yeah.
exactly.
Yeah. Yeah.
So are there some other examples in the literature, like what fair advantage TFs have got, so that it's this? For example, it can be done in RBPs, like RNA binding proteins, which are
Sure.
nowadays,
Sure. Yeah. Yeah. Yes. Yes.
or it is very exclusive to the TF itself.
So we don't. We focused only on transcription factors because this kind of RNA binding protein type, those types of proteins are not as they're not in large numbers. Excuse me. So not in large numbers in.
I think I lost him. Guys, anybody can?
Yeah, I think Aswin froze for a moment. Let him get back.
Yeah,
I thought I got frozen.
I also got like shocked somehow.
Okay. So his presentation is still on. But let me check with Aswin. Mhm. Aswin, can you hear us?
Dibarga, if you have his number, can you give him a call?
I'm
Yeah. And I don't know if there is this kind of data for
we
say.
lost you for about 1 minute. So
Okay, okay. So where was I?
no, so yeah, so you just were answering Gaurav's question. Gaurav,
So this is about the RBPs, I guess, right?
Yes,
Yeah, yeah.
RBPs.
So
Yeah.
I understood. So you said like no concrete analysis been done. If I
Yeah, so the problem also is, are there equivalent data available? So here, for example, for these transcription factors, I know some targets. And but for eukaryotic RBPs, do we know targets for a large number of these? I don't know. But if the data are there, then I don't see why it cannot be done.
Okay, okay. So, I mean, quickly, I think I have last question. So, Ashwin, I mean, so within your TFs are their targets comparisons. So you have seen some sort of effect. So,
Hmm.
is any bias for like operons? Because I mean, in E. coli we have like this polycistronic stuff. So,
Correct.
are there some bias like for those TFs which are very biased for the longer promoters, like longer operons or something, or it is random, it's
So
stochastic?
we didn't see any of that. We also, while we doing controls, we with things that were done were to make sure that we're not seeing something just because something has a large number of targets and we are averaging and all of that. So we didn't see anything. The only thing is, if you have more targets, then you have even less sequence variation.
Okay,
That's
got
it.
it.
Nothing to do with operon length itself that I remember seeing.
Okay, good. Thank you, Ashwin.
Okay, so this second part will also have some experimental things, really similar to the questions that came just now. So here I'm looking at this interesting phenomenon called long-term stationary phase, and the work that I will talk about. So the first part is done by Farhan and Sabita. Sabita was a postdoc in the lab. Now she's managing one of NCBS's field facilities in Pachmati. So she's enjoying herself there. Povitro is also a PhD student who did the experiments that I will talk about towards the end of this part. Now, with any with your biology in school or college, you did. Seen that when you take E. coli and put them in fresh media, they have a short lag phase where the cell numbers don't really increase, and then there is an exponential growth, and then after that there is stasis. There is it's probably hit carrying capacity, and this goes on for a few days, and then after 2 to 3 days there is a crash where about 99% of the cells die. So this is the typical growth curve that we do. Now, about in the 1990s, Roberto Coulter's lab at Harvard explored what will happen if these are maintained in stationary phase for a long time. Many bacteria and maybe even yeasts often they like to go into dormancy, or they even form these they sporulate and so on. But E. coli, what they found is that even though the cell numbers overall remain fairly constant over time, and but what one has is that this is a very, very dynamic phase where many different subpopulations of bacteria arise, and then they lose out, they die, and then some other population arises. So this fairly stationary phase is comprised of many small exponential growth curves. And this is the model that is proposed. So I may have a fairly constant cell count, but I might have one variant which is doing this, growing up and then dying, and then as it dies, maybe a different variant emerges, and this goes on as the medium composition keeps changing. And here the medium composition is changing purely from the activities of the bacteria that are there. And over this time, novel phenotypic forms emerge. For some reason, this interesting phenomenon has not been explored much outside of Stephen Finkel's lab in the last 15 years or so. But still, this promises to be a great model for studying evolution, fairly competitive and in a dynamic environment. What novel genotypes emerge? So that's just a simple what question. And then how does genetic diversity change over a time? And what kinds of genetic changes appear? Now, the first mutant that was isolated by Roberto Coulter was called ZK819, and it had carried a mutation in a gene called RPOS, which is a key protein which helps the bacterium transition from rapid growth to tolerating stress. So this, when you have active RPOS, also called sigma S, when you have active RPOS or sigma S, the bacterium favors stress response over growth, and it's the other side of this story is what is called RPOD or sigma D, which is a transcription regulator that is responsible for a lot of gene expression for rapid growth. Now they had, yeah.
So, Ashwin, sorry. So, is it the sigma factor for the RNA polymerase?
Yes, these are RPOS and RPOD are two sigma factors. RPOD is also called the major sigma factor, responsible for housekeeping functions as well as, which includes all kinds of nutrient utilization. RPOS or sigma S is the stationary phase or stress responsive sigma factor, which activates the general stress response. And these two compete for binding to
Polymerase.
the RNA polymerase. Yeah.
Got it. Thank you. Thank you.
So this is Roberto Coulter's mutant. This is RPOS mutation, which in fact has an attenuated RPOS activity. And we started with this, and then we basically sequenced populations as they emerged over a 1 month duration, where E. coli is just maintained in stationary phase with no addition of new nutrients. Now here we're just showing some genes in which mutations were found over time along this x-axis, 4 different lines that were evolved along the y-axis. Red means that the mutation is present in at least about 60% of the sequencing reads. Gray, mutation not found. Low intensity red, low prevalence of this mutation. So here you see that there's no mutation that we observed that had been fixed in the population, that had never reached 100%. And throughout you see, as in line with this model, you have variants emerging and then disappearing. And there are very diverse phenotypes can be seen. And over time, the genetic diversity of this population increases, which may increasing complexity of the medium composition, or the multiple mutations that might exploit the same niche, or just even maybe random drift as well might happen. So to look at this, we looked at first what kinds of genes are these mutations in. And getting back to this theme of mutations in transcription regulators, we found that many mutations, I don't know if it can be seen clearly, those are marked in a blue color in this figure. So these are all mutations in regulators of transcription. And in addition, these proteins which are marked by these arrow are mutations in the RNA polymerase itself. So these are all mutations which can directly affect transcription of a subset of genes. And where do these mutations occur in these proteins? Often these mutations target residues which are highly conserved, and many of these mutations also result in physicochemical switches in the targeted amino acids. So what we are seeing here is that evolution in stationary phase produces exceptional genetic diversity. This is fairly large. I didn't show this in detail, but the level of genetic diversity that we see is much higher than what we would expect by random drift. Many mutations are seen in regulatory systems, which is something that we like, and very likely that this is under fairly strong selection. Now, again, also pointing to the question that was asked, we wanted to look at some of these mutations in detail and see what they are doing. So what did we choose? We could have picked any, but we had to choose one. We can't look at everything. A student can probably do 1 or 2 in his or her PhD. So we thought, okay, we know that there are different colony morphologies, cell morphologies emerge during these long-term stationary phase, and then we saw that about 3 to 4 weeks into stationary phase, these colonies, which are very small. So in this image in the black background, you see these larger colonies, which are a typical E. coli colony, and then you also see these very, very small colonies. So we decided, okay, let's look at these small colonies and see what mutations they have. So these are small colonies, which also means they are slow growing variants. They grow slowly. They have long log phase. Okay, so can they do they show any selective advantage in stationary phase? So we need to do some basic phenotypic characterization. Now this mutant seems to, if you look at third, so this should be third row, first column, where you see the scissor type pattern, where the mutant is starting in minority and the parent is starting in 1,000-fold majority, and then after a few days of competition, the mutant is overtaking the parent. And this is also happening only in a very limited time range, because this is a fairly dynamic environment, and we wouldn't necessarily expect the selective advantage to be maintained in many different conditions. So this seems to happen only in conditions where the media has been spent with E. coli for about 10 days or so. So this variant, this small colony variant, can outcompete its parent in stationary phase, but in a very narrow set of conditions. And it can also outcompete its coexisting variant. So in this case, we've competed it with the original parent that we started the experiment with, which is something this variant may never see, given that this variant emerged only some 3 weeks after the experiment. Now we competed it with its coexisting large colony variant, and again we find that this wins in this competition. Okay, now we need to characterize what mutations these have, and we found that there are two different mutations that distinguish these variants. One is a mutation in sigma S, which we've talked about, that is the sigma factor for stress response. And we also saw a second mutation in RPOC, which is a core subunit of the RNA polymerase itself. So two different mutations which distinguish the small colony variant, the coexisting large colony variant, as well as the parental RPOS 819. So that was called. Now, what RPOS variants do we have? So the starting parent has this RPOS 46 or RPOS 819, which is attenuated activity. Now, this SCV has an RPOS 92, which is a variant which we call it as RPOS 92, and the large colony variant has wild type RPOS. And we know that RPOS 819 is an attenuated RPOS. That means your stress response is in fact down-regulated at an early stage in this prolonged stationary phase. And what we see, we thought, okay, maybe this would be a theme that would go, that would extend into very, very prolonged stationary phase, and that RPOS 92 would probably show a stronger attenuation of activity. But what we saw is in fact the opposite. At later time points, RPOS mutates again, and it seems to mutate so that it can revert back and achieve an activity that is closer to an wild type status. So at early in prolonged stationary phase, it looks like RPO, the balance between growth and stress response is in favor of growth, and at a later time, it seems to shift back towards favoring stress response. Okay, now we want to start to look at the other mutation, the RPOC mutation. What does it do? Again, these become problematic because there are multiple mutations. But we can, this is E. coli, we can do some genetics in using a bacteriophage transduction to put the RPOC mutant in various RPOS backgrounds, and also revert the RPOC star back into a wild type background, and then characterize these the effects specifically of the RPOC mutation. And so there are two areas that we can focus on to looking at this RPOC effect. One is the slow growth phenotype; it grows slowly, and the second one is the ability to give a selective advantage in late stationary phase. So and both of these are things that we can look at in different backgrounds of RPOS. Now, the first thing we see is that this mutation, the RPOC mutation, does confer a slow growth irrespective of the status of the RPOS in these strains. Whatever we do, it is a slow growing mutant. And why might that be? Some experiment trying to induce transcription in these mutants show that RPOC doesn't induce transcription very well. So it's just a slow transcription. A poor transcription variant, which is probably why, irrespective of what RPOS is doing, this just doesn't transcribe very well and therefore results in slower growth. Now, the second question: does it confer advantage in stationary phase? Now, this seems to be very strongly dependent on the status of RPOS. Now, it does confer advantage in stationary phase, but it does so only when RPOS activity is at a low level. That is, if RPOS activity is approaching wild type, then it really doesn't have any effect. And if there is no RPOS as well, it doesn't have any effect. So it needs a low level of RPOS activity to be able to confer this growth advantage in stationary phase. And it seems to be operating through RPOS because the delta RPOS strain cannot give this advantage. And so if we do the transcriptome of this RPOC mutant, and we find that it seems to be selectively upregulating targets of RPOS. So there seem to be multiple ways by which E. coli seems to be reactivating RPOS very late in stationary phase. Therefore, by finding multiple mechanisms to switch the growth stress response balance in favor of stress response. To summarize this, we have a mutation in a core component of the RNA polymerase, which activates the general stress response when you have a low RPOS activity. You need RPOS also for this, and this causes a GASP phenotype. And the RPOS activity seems to be changing in deep stationary phase. We do not know how this will pan out if we extend beyond the 1 month that we've looked at. Would stress response that's activated now be a requirement for a long time, or would it switch back at some point back to a situation where it would favor growth? These are questions that would be interesting to answer, but we don't have an answer to. So thank you very much.
Hi, Ashwin. Thanks for the great talk. So anybody in the audience, if you have any questions, please ask. Okay, anyone among the students? All right, okay. So I think Gordon's asked a lot of questions. So one thing to ask Ashwin, in the study that you referred to that has about 60,000 generations of evolution for E. coli, do you have expression data as well there, or it's
No, they've studied expression for some lines here and there. Not for everything for which we have genome data. There is, I think, Dominic Schneider's lab in Grenoble. So, who I think he was a postdoc or PhD student with Richard Lenski in the past. So he has been looking at certain lines that came from this study, which had certain mutations in global transcription factors. So he's looking at those in some detail. So yeah, there are individual lines for which gene expression information is available, but not broadly, to my knowledge.
Got it. Got it. All right. So the other thing that I wanted to ask was, I have been working on embedding mutations, wherein, because the mutations are very rare. If you look across the population, then the chances of having the same mutation across multiple individuals is very less.
So
So,
this is in what organism?
I'm doing it in humans.
In humans. Okay, okay.
Yeah. But I was just wondering, what is the situation with E. coli, for instance? Is it such choppy, or it is much more dense? If I look at the matrix of the, uh, you know, different, strains and their mutations, is it a very dense matrix, like you have multiple instances of the same mutation occurring, or it's very discrete?
So we have fairly interesting data from these, especially from these lab evolution experiments. And many of these lab evolution experiments have shown quite a bit of parallelism, where mutations in the same gene, if not the same mutation in the same gene, do show up again and again and again in multiple lines, which is indicative of very, very strong selection. So there is a lot of parallelism. So one can get
So
the same.
you are saying the mutation has not to be the same, but it's often on the same gene. Of course, you have not a lot of genes in E. coli, so you have a lot of co-occurrence on the same genes.
No, this is well, we're still dealing with 4,000 genes. But I would guess that many of these would, where you have multiple mutations in the same gene, many of these would be loss of function mutations. Because I guess it's just a chance that you can mutate, you can have the same effect with the multiple mutations. I think where you can have a loss of function doing the same thing, you do see these.
Got it.
For example, if you look at this RPOS, which has been attenuated in a variety of different studies, most of these conditions pose the problem of prolonged slow growth on this population. And in all of these situations, they found all kinds of mutations in RPOS, and pretty much all of them are attenuating. So I guess it's, with loss of function, it's possible.
Yeah, the other question is from the first study where you described that the mutations and the transcription factors are. I think there's a noise from somewhere. So
Yeah, I can't hear properly. There's some.
just wait a second. Let me see where it is coming from.
It's better now.
I think it was coming from Gaurav's side. So I put himself on mute. Hi. Okay, back to the question. I think, with reference to your first part of the talk, where you demonstrated that the transcription factors are, on the contrary to the RDR reports, it's more conserved, right? In case of E. coli. Yeah. Now the thing is, why it is more conserved. I didn't understand that. Did you get into why? Like, you talked about some horizontal transfer and all that, but
that comes much later. So,
right.
So the reason we think that mutations in these transcription factors would be selected again would be just because these mutations are likely to be pleiotropic, and when you have adaptation via a mutation in a transcription factor early on, it's almost like carpet bombing. So it's a single mutation which can probably affect a large number of things, and probably adaptation to these conditions would also require more than one pathway to be altered. But presumably as time goes on, and selection would find these more specific mutation. Say it's affecting 100 genes at the beginning, but you really need to change only 2 or 3 for it to be to get a selective advantage. These 3 maybe can be found over time, and as that happens, this big carpet bombing thing that happened early on would start to lose out. So it's likely that those mutations would now lose out in population, which would mean that transcription factor mutations would be selected against. That's the best explanation we have.
Got it. No, that makes sense. Sorry, I guess anybody, if you have any question, you can pitch in. Yeah.
So Debarkar,
I had a few other questions, but I will come after Sriram. Yeah.
Yeah. So it's a very naive question, no? Like any of the evolutionary changes that occurs at a very long time. Any new phenotypes actually that which generates actually, it's basically the survival of the fittest,
Right.
that which occurs over the period of a very long time. But in your case, what you are saying, even in the E. coli, the evolutionary changes seems to be occurring at a very fast. I know that the lifetime of a E. coli or the replication stage is something like 40 minutes to 60 minutes,
Yeah.
and the new generation keeps coming actually. Of
Yeah.
course, 40 and 60 minutes are the record time actually you can think of. But is it not something very contradictory that like it is not happening at the usual evolutionary time scales, and it's happening at a very small time scales, right? In
It,
the in
it,
the in the usual sense, right? Like so, how do you reconcile to that fact that like it's happening so quickly when we
it,
talk about the evolutionary time scales as basically extremely big?
No, because you reconciled it already when you said that the generation times are whatever 30 minutes or so, and also the population sizes are very very large, right? So the chance that you will find a mutation that gives a selective advantage is pretty high. For example, a classic example that one would talk about is resistance of E. coli to the antibiotic rifampicin. If you put you put E. coli to sublethal concentrations of rifampicin in the lab, within 24 hours it will be resistant, and the resistant population will take over. And it's just that there are enough
But,
cells,
but
and you
that
can
is
find it.
basically but that occurs basically due to the presence of plasmids, right? That like you're
No,
talking
no, no,
about
no,
the
no, 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 no,
transmission.
no, 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 no. This has got to do when I'm talking about this in the lab evolution situation. You're talking about only mutations without the chance of a horizontal transfer, and these would be rifampicin mutation resistance. In these cases, would emerge purely from mutations in the RNA polymerase itself, in the beta subunit.
Okay.
Um, and even with antibiotic resistance, though a lot of what is clinically relevant in, say, something like E. coli, and for many of these would be horizontally acquired plasmids and so on. For Mycobacterium, many of these are point mutations. And
Oh,
this
okay.
just
So
happen
yeah,
very
I was
fast.
about to come to the question basically like. So I was about to come to the question like, what is the type of mutation you're talking about? Because you are very generally talked about your mutation. What sort of mutation is that? Like, have you categorized the type of mutation that was happening there actually?
So these are all non-synonymous point mutations.
Okay. Okay. Yeah, and that's fair enough. Like, second question is that like you about your second part of your talk.
Mhm.
In the last slide, you talked about basically the stochastic stochastic fluctuations, right?
I'm
Uh,
not sure it's
uh,
stochastic. Not sure it's stochastic. At this uh, and that it is switching from growth favoring to a stress favoring situation, and we probably need to extend this for much longer time scales to see if it would revert back. That I don't know.
Yeah, because you talked about Adam Arkin's work, and he was the first one to talk about basically the role of stochastic fluctuations in getting the various types of phenotypes, right? Like how actually the stochastic fluctuations ensures that like it's a phenotypic switching may take place, right? Like
Sure, sure.
so something like this. So, what will be your opinion that if suppose that you take these stochastic fluctuations into consideration, will there be will there be that the phenotype which is there in the stationary phase over the period of time that will be stabilized and overcome basically the original phenotypes that which it started with, right? Like so, is there a possibility there something like that?
So we don't know for over, because the biggest problem with the stationary phase stuff is fairly difficult technical challenge in that we do not know what is happening in the medium. We do not know how the environment is really changing, And
But I thought so. You are keeping the medium constant, right?
but it's
So.
we are keeping it constant, but the bacterial metabolism is changing the medium in its own way, and that we don't know how,
Okay.
because
The
there
composition is changing actually.
yeah, and then the composition changes. The first population goes up, it dies, it probably lyses, it releases whatever it is producing out into the medium, and that is changing the selection pressure towards something else. That's the problem, and we have no idea whatsoever what's going on there with
So
the,
what is the so in that case then what is it possible for you to reproduce the same type of phenotypes as we actually do it for the multiple replicates when you are doing it?
So with this, so when we did these four different parallel lines, we again saw several mutations coming back again and again in multiple populations. So there is parallelism in this. And also, for example, the small colony phenotype, though they may be genotypically different, but presumably their higher tolerance to certain stresses seems to be also a common theme which is coming again and again. So given the same starting conditions, it looks like there is reproducibility. And Steve Finkel's lab has also shown that you can switch these to a different set of mutations if you're changing the initial conditions. So in this, there is some level of determinism
So
in the selection that is operating.
so it's something behaving like a Candida species, right? Like it can go from one particular type of phenotype switching to another switching. If you do change basically your stress conditions or something like that, so that the one transcription factor will become inactive, the other one will become active, so that the phenotypes switching may take place, right?
Exactly. So here we're looking at something very similar with switching between growth versus stress response, which is a very classic phenomenon.
Okay. So one last question is that the stress response you are equating along with the selection pressure, right? Is these two are synonymous for you, right?
No. So, stress can be a selection. Stress response is what the bacterium does to respond to the stress.
Because I keep hearing that when you are talking about when you are presenting, I heard stress response as well as the selection pressure. So
Yes, they're different.
I was not very sure that whether your stress response is creating a sort of a selection pressure for the new phenotype to emerge.
So I would say stress response is the response to the selection pressure, which presumably is some kind of stress. And since we don't know what the medium is doing, we don't know what kind of stress that is. So the selection pressure comes from the medium. Stress response is the way the population or the cell responds to that selection pressure and
Yeah.
activating the
Thank you a
stress
lot. It was
response
very
itself
nice
as
talk.
well. Thank you.
Yeah, thanks a lot. It was a very nice talk and it was very nice. Okay, Debarka, you can take over.
Swin, I have a very funny question, and this is because of my poor knowledge in biology. And see, I mean, when you talk about mutations and evolution, it's basically it's a progressive term, right? I mean, things are adapting and stuff like that, and responding to the competition, the stress, stuff like that, right? I mean, but is it a forward-looking process all the time, or I mean, for example, why the mutational burden should only increase? They can also decrease, right? And it can go back as well. So the question is, all these mutations which are happening, are they in response to their competition with each other, within the strains? Or okay, or it could be that they are just relaxing. They can, if I, for example, if you create a more favorable condition for them, then what will happen to the mutation? Just a hypothetical question. So instead of creating stress for them or creating competition for them, if you remove competition, right, and you remove stress, then in which direction the evolution progresses?
So again, for example, in the mutation that I talked about, the small colony variant, it does very poorly if we put it back in fresh medium. there's no way it's going to outcompete a wild type. So we haven't done that, but we thought we would do it. But I don't know if we will do it. Is that you, as you put it, back in fresh medium? This is not a happy mutation, and it's going to lose out, and you're going to probably find either a reversion of this mutation or something else that compensates for this increased
Oh, is
stress
it?
response.
Okay.
Right? That's probably what's going to happen. And some of the other mutants, which we didn't really characterize in detail, when we put them back, which came up in these long-term stationary phase type things, didn't really differ from the wild type much in fresh medium. So I think there are all kinds of things, and depends on the particular mutation, and it may either do worse or not really care if we put it back in fresh medium. But again, the selection pressures would be very different there.
Got it. Got it. No, that's really intriguing. Anybody else wants to ask any question, or we are anyway 15 minutes overshot the time.
No, I think Dibarka, there is one guy named Gaurav. He wrote something on the chat option.
Oh, is it? Gaurav, so you can speak up then.
Ashwin, somebody has written, I don't know who's this person, but sir, one of your paper, he unquoted your paper like evolutionary analysis of bacterial non-homologous and
Oh,
joint
yes.
pair.
Yes.
So, which was published back in 2019. You wrote about two proteins, Ku and
Yes.
LID. Yeah. So the questions are as follows: were these proteins were mutated? Second is, where the data of sequences of proteins used from the lab evolutionary experiment retrieved from NCBI?
Yes. These were all from NCBI, and well, there is. If we're looking at these proteins from two different organisms, they're not going to be identical. So there is that natural variation in these sequences. So this one, again, we weren't looking at the kinds short level mutation spectrum. Our interest was more in trying to find out how this repair system, which is pretty much ubiquitous in eukaryotes, but a sort of sporadic occurrence in bacteria might have emerged. So that it was a completely different question. But all of these were just obtained from NCBI. You can get it from NCBI, EBI, KEGG. It'll be the same data.
So I think Gaurav, whoever Gaurav he or she is, so you got the answer from Ashwin. Thank you, Ashwin.
Thank you.
Ashwin, thank you, Ashwin, for patiently taking up all the questions. It was an amazing discussion. We learned a lot of things, and thanks again for accepting Rough Talking in the webinar. With that, I think we can all break out. Thanks, Ashwin. I'll catch you later over phone. Thank you.
Yes. Thank you. Bye.
Bye.